Okay, so here's the video on cells. This one's been kind of condensed, and I'm going to have to split it up a little bit just because there's so much. For the organelles, that one, I don't really want to write them all out because you have that PowerPoint that has all the review stuff on there. So just look at those, worry more about function instead of structure for those ones. So the first part, prokaryotic versus eukaryotic, these are like exact opposites of each other. Prokaryotic, small and simple, no membrane-bound organelles, no true nucleus, and their example would be bacteria. So if you think about them, you are eukaryotic, just compare yourself to a bacteria. You are bigger and more complex, you have membrane-bound organelles, you have a true nucleus. Animals and plants are the ones that are eukaryotic. So they are exact opposites in all those ways. Cell theory, 3 parts. All living things are made of cells. We talked about this for characteristics of life too. They'll have to have at least one cell to be considered living. They're the basic unit of life, meaning that life does not start until the level a cell is reached. So things like organelles, molecules, atoms, those are not living things. All cells come from other cells, so they have to come from some sort of pre-existing cell through some sort of cell division. Careful here, because some of you said like all cells are made of cells, and that doesn't really make sense. So just, I mean, this will be recognition, so you don't have to worry about that part, I guess. For isotonic, hypertonic, and hypotonic solutions, I have some examples down here. Just really quick for the vocab. Solute is what gets dissolved, solvent is what does the dissolving, and solution is the solute dissolved in the solvent. So you will see another one of the things like this. You've already seen these before. The first one has 5% salt outside, 20% salt on the inside. So there is less solute outside. That's how we label it hypotonic. I'll ask you to show me the movement of water. If you forget how to do this, if there's 5% salt, that means that there is 95% water. If there's 20% salt, that means that there is 80% water. Naturally, water always wants to move from high to low, so it'll go in. Again, this will be multiple choice, so you won't have to worry about drawing your arrow. You just have to recognize which one's correct. But just to reiterate, some of you are drawing arrows that don't even go into or out of the cell, like they're next to it or something. So make sure you're showing me that it goes from the solution into the cell, so one of those ways. The next one is isotonic. Iso means equal, so there is 5% salt outside, 5% salt inside. They're equal to each other. That means that there would be 95% salt outside, 95% water outside, 95% water inside. So they're going to move in and out at the same rate. So you can write no net movement, or you can do one of those arrows kind of going in and out, showing me that they're coming in at the same rate. The next one is hypertonic. Hyper means more, so there's 5% salt outside, only 1% salt inside, so there's more solute outside. Again, to show the arrow, you calculate how much water is there. So 5% salt means there's 95% water, 1% salt means 99% water. Naturally, water wants to move from the 99 to the 95, so it goes out of the cell. Calculating the water is probably your safest bet, but if you can remember, hypo always gains, hyper always loses water. So if you're able to remember that, then that's fine too. For the transports, there's 2 types, passive and active. Passive transport always goes high to low and never ever requires energy. There are 3 types we talked about: simple diffusion, small nonpolar molecules that just move through the phospholipid bilayer, they can just go straight through. Facilitated diffusion, these are large and polar molecules. They need help of a transport protein, so the phospholipid bilayer is semi-permeable, meaning that it only lets certain things in and out. That's a word you need to know. So some things have to go through some sort of transport protein. They're really specific for what they let through, but they offer little channels or little like clampy things that are going to let the molecule get through. And osmosis is always only going to be talking about water. Nothing else can do osmosis. So all 3 of these move from high to low, never require energy. That's how they're all the same. Active transport, they all require energy. Active transport, the actual act of it, so remember it's a category and a type. So active transport is any molecule moving from low to high. Endocytosis is any molecule that gets engulfed by the cell membrane and into a vesicle, so it goes inside. Exocytosis is the exact opposite. Something is secreted by that vesicle fusing with the membrane, kind of kicking it back out into wherever it's going to be.